Hey everyone, I'm just gonna introduce myself real quick and tell you what I do for Children's Hospital of Philadelphia. My name is Alex Lubneuski. I work for Data-Driven Discovery Center in Biomedicine in Children's Hospital of Philadelphia Research Branch, and I lead cloud infrastructure for the center. And Jeremy, why don't you introduce yourself?
Yes, of course. So I'm Jeremy Costanza. I'm working at the Saint-Justine Children's Hospital in Montreal, and I'm a tech lead developer.
Great, awesome. So let me tell you a little bit about ourselves. So we started about 8 years ago at D3B Center, and our center is mostly uh, centralized around researching cancers and brain tumors, and pediatric cancers and brain tumors. And we're building tools, collecting data, we share data between the researchers, and that's our main goal in the research community. Jeremy, do you want to say a couple words about Saint-Justine?
Sure. So on my side, I'm part of Fairlab, a unit of the research center at Saint-Justine Children's Hospital in Montreal, and we build applications to improve healthcare and make research easier, especially in connection with genomics.
Perfect. Okay. And so what do? We collect genomic data from multiple patients, de-identify them, and share items with the world for the research purposes. We additionally build and provide tools for researchers to conduct in-depth research in genomic data. So over a few years, we worked on several projects. One of them was a Kids First project. It's one of the big projects that was funded by NCI. And the point of that project was to collect as much data from the patients as possible, which would be actual tumors, and sequence them, and then upload them into kind of central locations so we can share between multiple researchers, provide them tools so they can do more in-depth analysis, and most importantly, to simplify access to the data, because before it was quite cumbersome. And the second project that we're working currently is Include DCC. So the first project is mostly for pediatric tumors and cancers, and Include DCC, it's also genomic data, genomic phenotypic data, but it's for the children with the Down syndrome. All right. So before we're going to talk about our infrastructure and data structure and all the good stuff, I feel like we need to talk a little bit about Genomic Primer. So we'll talk about genomics and the terms that we're going to use in this presentation. So we collect whole genome sequencing for patients and participants. And so whole genome sequencing is an experiment made on a person to sequence the whole genome. A genome is made of 3 billion pairs, positions. Someone might remember from 8th grade biology class. Each base can be one of these, A, T, C, and G. Variance is one of the important terms that we need to learn about. Variance is the difference in DNA sequences between the reference genome and a different individual's DNA. So as we know, 99.6% of genome of all people is identical, and there's about 0.4 difference. And so scientists created a sort of a template. It's called the reference genome. And so that reference genome is really useful when you're comparing the patient's genome with a reference genome, and you can see what the difference is. And that difference is called variance. So there's two types of variances. So there's semantic variance, somatic variance, and germline variance. So germline variance is the one that inherited, and the somatic one is not. I put the link to a reference genome, and anyone wants to learn more. So another term is occurrence. Occurrence is a specific instance of identified genetic variant in the patient associated with particular sequencing experiment to run. So as we talked in the beginning, whole genome sequencing is an experiment. So sometimes what happens, you do whole genome sequencing, and then you see the variants, and you're interested in a specific part of the genome sequencing. So you take that part, and you do a panel, what it calls. So it's also sequencing, but you do other type of experiments. And so if you see that variance in those two experiments, that's how you count the occurrences. So there'll be 2 occurrences if you do that twice. We also captured a couple metrics. One is zygosity. It's whether a patient has a variant in one chromosome copy or both copies. Depth, it's how many sequencing reads covered the genomic position. So when you do a whole genome sequencing, it's really hard to do it from position 1 to position 3 billion. So the way it works, you will split it into random pieces, and then some of those pieces to overlap. And sometimes they overlap, 10 times, 100 times, 1,000 times. And so every time it overlaps, it basically rereads the same position. And so the more times you reread the position, the more certainty you have that you have a specific letter in that position. And also we care about genotype quality. It's a score indicating how confident the sequencing technology is about to recall the genotype. So this is our model, a very simplified model. So we already talked about variance, occurrence. There's a couple other tables that there. There's a ClinVar and HPO. So the HPO, what's important about it is basically connects the position in the genome with a specific term for a disease. And the ClinVar is a public database maintained by NCBI, and that aggregates information about genetic variants and their relationship to human health and disease. And there's a consequence table that we, like the consequences that we noticed with a particular variant. All right. So before we stumble upon StarRocks, we used OpenSearch in our both of those portals, Kids First and Include. The architecture is pretty simple. We have UI, we have a few microservices, and the OpenSearch cluster. Additionally, we had a genomic data lake. It's we AWS based, so it's a glue tables. And so we used ATL, like a big Spark cluster on the EMR in order to denormalize the data and push it into the OpenSearch. And so the way we did it, we created a new index. We pushed that data into the new index, and then we switch it up between the old and new index for the API to reach. And it was great. It worked really well for what we currently have. The searches are really quick, so it's because Elasticsearch and OpenSearch. It's built on Elasticsearch. The reads are really quick. API is great, well documented. It is open source, it's important to us, and it's easy to install to maintain, especially open source since it's a SaaS product. The issues with OpenSearch is normalization, dolomization of the data. So that obviously creates some issues with redundancy. OpenSearch writing is very inefficient. And it's also inefficient in storing data, which I already mentioned. We basically, when we denumeralize it, our data become a lot bigger. And obviously, there's no joins in OpenSearch. And querying data in OpenSearch is not easy if you have a nested relationship, which we do have a lot. And Jeremy will talk about the new project we're working on and how we use StarRocks there. Go ahead.
Thank you, Alex. So Radiant is our new project. So what is Radiant? Radiant is a multi-institutional program funded by ARPA-H and led by Children's Hospital of Philadelphia. And my team at Ste-Justine received funding to take part in developing the solution. So here the main feature of Radiant. First, Radiant will collect multiple types of datasets from healthcare system within each institution. We are talking about genomics, clinical reports, surgical notes, imaging, and so on. And we're looking at about 75 organizations in the future. Next, Radiant will give clinicians a tool to analyze data at the patient level, enabling what we call precision medicine. I'll come back to this in a moment. And finally, institutions will be able to securely share anonymized data with each other That way you can do things like search for patients similar to the one you are analyzing. Next slide, please. So let's take a look at some key differences between Radiant and existing portals we developed in the past. First, Radiant represents a shift from research focus to clinical grade application. Previous projects like KitFirst or Include were mainly used by researchers to find common variants across whole genome sequencing datasets. Radiant takes this step further. It's used not just by researchers, but also by clinicians to help establish diagnosis and support precision medicine. So what is precision medicine? Basically, precision medicine means customizing treatment and care to each patient based on their genetics, medical history, and other personal information. Then clinicians pick the treatment that works best for that specific patient. About deployment and infrastructure, past projects relied on a single large Elasticsearch cluster per project. Radiant is completely different. It is completely decentralized. Each organization is responsible to manage and deploy its own infrastructure. We are targeting around 75 organizations, so we need the application to be easy to deploy and maintain. About update frequency and data freshness. So in previous project, updates were batch oriented, occurring 3-4 times per year as new study data became available. Radiant needs to be much faster. Ideally, updates happen every time few whole genome sequencing are available. That means daily or weekly. Sequencers usually produce whole genome sequencing in batches of few dozen. And this is related to the last point of the table, criticality and ingestion duration. So for KitFirst and Include, ingestion was not critical. Taking a couple of days to process data was acceptable. For Radiant, speed matters. So there is a patient potentially in a critical situation waiting for diagnosis. So ingestion must be fast, ideally within few hours. Next slide, please. Let's take a look at some key numbers to understand the impact of Radiant. So before Radiant, we were storing 500 million variants in Elasticsearch, one document per variant. Each variant could have multiple consequences, in average 2, plus aggregated information like how many patients had it or how many were homozygous. With Radiant, we are moving to the occurrences level. That's 63 billion occurrences, each tied to a variant and its consequences. So Elasticsearch just can't handle this scale efficiently. Why? Because to query occurrences there, we have to duplicate variant information for every single occurrence. That means much more disk space and slower updates. So Elasticsearch is a great application for search, but it's not built for billions of relational records. So let's see how StarRocks help us. Next slide. First, our tables are normalized, which not only saves space, but also speeds up ingestion time. Yeah, it's also possible to update just certain tables in a granular way, like the public tables Alex showed in grid, ClinVar, HPO, and so on. And it's possible because StarRocks shines with fast jumps, especially collocate jumps. So collocate joins are a very important feature for us in StarRocks, and it's probably one of the main reasons we picked StarRocks. For those who don't know, collocate joins let you define a collocation key. For many tables, rows with the same key will be managed by the same data node, avoiding shuffle when joining on the collocation key, and of course, making joins much faster. In Radiant, we chose for the shared data architecture. Why? Because it's easier to scale or replace nodes in case of maintenance. It also reduces cost by limiting the need to replicate data. So this is an additional advantage compared to Elasticsearch, since Elasticsearch needs to replicate data to ensure integrity. Here, integrity is ensured by the object storage, which is more cheaper. On the downside, with shared data architecture, performance can be affected, but when the data is in cache, it's the same as a shared mapping architecture. Another good thing about StarRocks is how easily it works with cloud services like S3 and GRU or Iceberg catalogs. So this is really helpful for ETL development, as we'll see in a moment. Next, StarRocks supports next. So StarRocks supports only SQL for queries. So we need to rewrite a new API. But with the maturity of the SQL framework, particularly Go in our case, because we are using Golang, development is pretty straightforward. And another advantage for SQL is universal, so you don't have to learn a whole new API to query StarRocks, which can be useful for other future use case like data analysis. Let's continue by looking into the architecture of Radiance. Thank you. As input, we have genomic files that comes from sequencer and bioinformatics workflows. We also have a clinical transactional database in Postgres. First, we process genomic files using the CS task operator in Airflow. It executes behind the scene Python code. So this task just converts genomic files into Iceberg tables. We configure an Iceberg catalog to create these tables in StarRocks. And we also configure a GDBC catalog over the Postgres clinical database. Next, the data gets loaded and the data gets loaded and transformed into StarRocks using a SQL operator in Airflow. So essentially, it's just queries that the bunch of queries doing insert into select statement. So what does it mean in practice? It means the StarRocks cluster handles the transformation itself. We can do it all in StarRocks, and no extra software like Spark is required in the infrastructure. So deployment and operations are easier for each organization. Next slide now. So let's have a look on performance, starting with the right side performance of StarRocks. So we set up a cluster with 5 data nodes in a shared data architecture. And we loaded the whole kit source dataset by batch of 100 whole genome sequencing. So 100 whole genome sequencing represents the amount we expect coming out of a sequencer. End-to-end ingestion for a batch takes about 2 hours. If we're breaking it down, parsing genomic files in Python and populating Iceberg tables takes 1 hour and a half. Then transforming the raw data into optimized table with StarRocks takes about 45 minutes. To achieve the same in our previous architecture, it would have taken more than a day. And so in conclusion, less space, less time, fewer resources, and more data. Next slide, please. So what about read-side performance? So we used Locust to simulate user sessions that are closest to the real-world usage. That means top-end queries, group by aggregations, of course, and joining tables. And we run this test with 12 concurrent users on the same 5 data nodes. And in short, as you can see, StarRocks delivers fast query even under realistic workloads. This concludes our presentation. Next slide.
Yeah, and one thing I just want to add that big thank you to Stellar Data for helping us with our POC, initial POC, and starting with the StarRocks. Well, thank you. Thanks everyone for attention.
Yeah, we certainly appreciate your partnership and your involvement, your contributions to the community. We're going to open things up. I know there's a couple minutes left. Are there any burning questions anyone has to ask? I know that we're kind of coming close up on time for the next session. If there are questions that we can't get to today, there will be opportunities to ask those questions in Slack, as Beryl has just pointed out in the chat. We will work with our speakers to get you answers to those questions. I do see something just came in. Thanks for your sharing. What were your freshness and query response time requirements? Was a question we just got.
Yeah, so for query response time, we target sub-second queries. So because people using our applications are geneticists or doctors, and they can't lost a lot of time waiting for queries to respond. So we're targeting sub-second queries. That's totally acceptable. And for data freshness, so I mentioned when data is output in sequencer out of sequencers, we expect between 2 or 3 hours to be in our applications.
Perfect. All right. Any other questions before we wrap things up today? Give people a couple more seconds to post something if there is something. Yeah, otherwise, this has been fantastic. I wanted to thank everybody for joining this session, but I especially, of course, wanted to thank both of our speakers, Alex and Jeremy. You guys did a great job. You're working on some very important and amazing things. Frankly, it's really great to see. Thank you again for sharing that. And to the folks in the chat, please don't forget to complete the poll question, and of course the QR code, scan it if you have the opportunity to, and join the StarRocks Slack channel. We'd love to see you there. But the next session should be starting in just a few minutes, so folks need to get to that. That's great. But otherwise, Alex, Jeremy, once again, thank you guys so much. This has been really fantastic.
Thank you.
Thank you.